Welcome to our video on Chapter 16 in Section 16.5, where we're going to talk about the ear. Our ear is made up of three different portions. We have the external ear that's responsible for hearing and will terminate at the eardrum or the tympanic membrane and will include structures such as the auricle and this external acoustic meatus, the canal here. We have our middle ear that is going to also be associated with hearing, and this is an air-filled space that contains these auditory ossicles, or the three small bones of the ear. And lastly, we have the inner ear that is going to be responsible for hearing and balance. And here we will see an interconnecting fluid-filled tunnel that is going to have chambers within our temporal bone that surrounds it. So let's first talk about all the details of the external ear. First, we have the pinna or the auricle, and this is composed of the helix, which is the rim of the ear, and the lobule or the ear lobe. Also, you see pictured here is our external acoustic meatus, or it's also known as the external auditory canal. And you can see that it's a short, curved tube that is lined with skin, and we can have our hairs, sebaceous glands, and ceruminous glands here. What are ceruminous glands? These are going to be modified apocrine sweat glands that lie in the ear, and they're responsible for producing earwax when it mixes with the sebum. A little bit more on the external ear. It is going to end at the tympanic membrane or the eardrum. So this is our boundary structure between the external ear and what we'll soon talk about is the middle ear. The tympanic membrane is made up of connective tissue, and it vibrates like a drum in response to sound. So it helps to transfer sound energy to the bones of the middle ear. Moving on to the middle ear, this is going to be that small air-filled mucus-lined cavity in our temporal bone, and it's flanked laterally by the eardrum, and then it is going to be flanked medially by a bony wall that's going to contain something known as the oval window. Okay, so oval window makes contact with the ossicle here. And we will also have a special area below that known as the round or cochlear window. In this area, we have a epitympanic recess, and this is going to be superior within the middle ear. And we have a pharyngotympanic tube, auditory tube, or sometimes this is called the eustachian tube. And this connects the middle ear over to the nasopharynx. So when you go in a high rise or are in an airplane and your ears start to pop, that's because your body is trying to equalize the pressure in the middle ear with the external air pressure. Now for our actual ear ossicles, or the three small bones here in the middle ear. First, by the tympanic membrane, we have the malleus, which is also known as the hammer. We have the incus, that is also known as the anvil, and the stapes, that is also known as the stirrup. And these are suspended by ligaments and joined by synovial joints. We have the tensor tympani muscle over here, as well as the stapedius muscle that can contract reflexively in response to loud sounds to help prevent any damage to our hearing receptors. Now for the bony labyrinth. So these bony labyrinths are basically chambers within the temporal bone, and it is made up of the cochlea. There we go. The cochlea that is involved with hearing. We have our vestibule that is involved with balance, as well as our semicircular canals that are also involved in balance. And we have our membranous labyrinth as well, and these are going to be the tunnels and chambers in the inside the bony labyrinth. Now, inside these chambers, we are going to have certain fluids. So we have endolymph, in the membranous labyrinth. And then we have perilymph that is found in the space between the membranous labyrinth and the periosteum of the bony labyrinth. Now it's hard to see in this illustration, so let's move into the next slide over here, where you can see an area of the cochlea has been sliced, so we can see a cross section of these chambers, and you could see that the light blue is representing the perilymph. Again, it's between the membranous and the bony labyrinth, and then in the center here we have the endo lymph that is found in the membranous labyrinth. Now let's try and put all of this information we've learned so far together. So we are going to have a vibration of sound waves move through the external auditory canal. That is going to make your tympanic membrane or the eardrum kind of vibrate, so that we can take those sound waves and transmit them through vibrations of the ossicles over to the oval window. Now, this is actually your cochlea. This guy over here, it is just unraveled. So this oval window is going to make contact with the vestibule, which is communicating with this special area known as the scala vestibuli. So here is a cross section of the cochlea. So imagine we just kind of sliced through here, and at the top portion here is the scala vestibuli. This is extending from the oval window all the way to what's called the helicotrema at the cochlear apex. And near this area, we will have responses to low frequency vibrations. The second cochlea chamber that we have is the scala tympani. So the scala tympani is going to be this region down here, again filled with the perilymph, and you could see the cross section over here. And the scala tympani can be found from this helicotrema over to the round window. So the reason we have this round window here is because we do need this fluid to displace just a little bit. The scala vestibuli and the tympani, once again, are filled with the perilymph in blue. Now, if you look at the wall of the scala vestibuli, you are going to see that we have a membrane known as the vestibular membrane. And if we look at the wall of the scala tympani, we have a wall known as the basilar membrane. If we look at this near the oval window, the basilar membrane is going to respond to high frequency vibrations. So remember, the low frequency was closer to the helicotrema, whereas the high frequency is going to be closer to the oval window area. We also have this region in the middle that is known as our cochlear duct, or the scala media. And this is the space essentially found between the vestibular membrane and the basilar membrane. And this area is filled with endolymph. So if you look, that is represented by the pink here in the center. So this is the scala media, or the cochlear duct in pink, filled with endolymph. And here's an illustration just showing you how that cochlea looks if we were to slice it down the side. So down here we have all of the scala tympanis. Here, 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 and here. Whereas the scala vestibuli would be the more superior region. Okay, here and here. And then the cochlear duct, you could see, is colored in that light purple or pinkish tone. Now within that scala media, you are going to see a structure known as the spiral organ, or it's also known as the organ of Corti. And these are just special cells within the cochlear duct. They're going to contain some hair cells there, which are sensory cells that look like hair-like projections. We have some inner hair cells. So here's a magnetized view of what that organ of Corti looks like. And you could see that we have some inner hair cells here. These are going to be responsible for hearing, and some of the outer hair cells are going to be there to regulate tension on the basilar membrane. And the basilar region of the hair cells is covered by synaptic terminals here that are then going to be afferent fibers that form the cochlear nerve. A quick clinical view here on otitis media. This is when we have an infection of the middle ear. Now, this is mostly experienced by young children because the shape of their auditory tube is more horizontal and shorter than that of adults. Usually this is due to a causative agent from a respiratory infection, and it may spread from the area of the pharynx through the auditory tube and into the middle ear. We start to see that fluid can accumulate in that area, and we have pressure, pain, and sometimes reduced hearing. So we can check this out by using a otoscope, and if it is problematic, then a myringotomy, which is a surgical procedure in which the tympanic membrane, pictured here, is going to be punctured and a tube is placed there, allowing some fluid to drain, and hopefully that otitis media can be relieved. So here is a full walkthrough of how a sound wave pathway moves through the ear. There is a linked video here that I really love. I'll place that on Blackboard for you to watch as well, or you can open up this PowerPoint and just click on that link. Anyway, what happens is when we have sound move into our ear, the waves are moving through the auricle into the external acoustic meatus and hit our tympanic membrane, causing it to vibrate, and then these ossicles will also become amplified and vibrate over toward that oval window. The stapes hits that oval window and generates that pressure to move through the perilymph within the scala vestibuli. Now you can notice as it's moving through this, through this chamber, we are going to have pressure waves that cause the vestibular membrane. Remember, that was the wall within the scala vestibuli to move. And this causes the pressure wave to shift into the endolymph within that cochlear duct or the scala media and displaces the basilar membrane that was the wall of the scala tympani. Okay, we got all of our names straight. And when it does that, the hair cells in the organ of Corti are going to be distorted, and that gives us the nerve stimulation that will be picked up by the cochlear branch and eventually sent over to our vestibulocochlear nerve, cranial nerve number 8, and will travel to the auditory cortex. Now, the remaining pressure of the waves are then going to move through the perilymph in the scala tympani and are going to be absorbed by that round window that is going to be displaced. So you'll see a little bulge out in this area. Now let's talk about the central nervous system pathway for hearing. So we ended up sending that sound information through this cochlear branch, which then is going to travel over to our cochlear nucleus, and then another neuron will take one of those signals over to the superior olivary nucleus, which then will travel over to the inferior colliculus. The other branch is moving from the cochlear nucleus directly over to the inferior caniculus. So from there, we are going to move into the thalamus and synapse at a specific nucleus, which is the medial geniculate nucleus. Remember, in the eye pathway or the vision pathway, it was the lateral geniculate nucleus. So make sure you know the difference and the specific nucleus that it synapses with in the thalamus. So from here, we are then going to travel over to with another neuron to our primary auditory cortex so we can interpret that sound. And the last thing we'll talk about is balance. So here we have our static labyrinth, which consists of the utricle and the saccule within the area of the vestibule. This is going to help evaluate the position of our head relative to gravity and detect linear acceleration and deceleration. So when you're sitting in a car, or maybe you get on one of those moving tracks at the airport. And then we have our kinetic labyrinth that is made up of our semicircular canals over here. And these are going to evaluate movement of the head in three-dimensional space. So let's talk about that utricle and saccule a bit more. Our utricle has a macula that's oriented parallel to the base of the skull, and our saccule has a macula that's oriented perpendicular to the base of the skull. So what's a macula? A macula is going to be specialized epithelium that is going to have supporting columnar cells and hair cells over here that are consisting of numerous stereocilia, which are just specialized microvilli, and we will have one cilium or kinocilium. So you can see that at the very top, it's this balloon-like structure. And this is embedded within a gelatinous mass weighted by otoliths. So oto referring to like the ear, and lith means stone. So these are like stones sitting here on this gel matrix. So this gelatinous mass will move in response to gravity bending the hair cells. So imagine you have this sheet that just shifts as you move your head. And then what that's going to do is initiate the action potentials to move through these neurons and move over to the correct area in the brain to interpret head position and acceleration. So here's a nice demonstration of that. Here is our macula with our head upright. You can see those hair cells are nice and straight, as well as the otolithic membrane with the otoliths on top. And then when we bend down to pick up a box, we see a shift in the head, right? So then we have a movement of that otolith and the membrane, and that is bending the hair cells along with the kinocilium, and we are going to send that information through these afferent neurons. Now for our kinetic labyrinth, remember that consisted of our three semicircular canals that are going to be filled with endolymph. So we have a transverse plane, a coronal plane, and a sagittal plane. And the base of each expanded will consist of an ampulla with sensory epithelium called a crista ampullaris. At the top of this crista ampullaris, we will have what's known as a cupula, and this is going to act as a float that will displace by fluid movements within our semicircular canals. So this displacement of the cupula is the most intense when the rate of our head movement changes. Thus, the system detects a change in the rate of movement rather than the movement alone. And here is a nice demonstration of this. This is showing us our crista ampullaris at rest. And then when this figure skater starts to twirl around in circles, that is going to displace the cupula, and we will understand where we are in space. Now let's talk about the neuronal pathway for balance. First, we are going to have the vestibular nerve communicate the afferent nerve fibers to create our vestibulocochlear nerve. And we are going to synapse here at the vestibular nucleus. From here, we will get signals sent over to the cerebellum that influences our posture muscles. And we will also send over axons to motor nuclei that have to do with our oculomotor trochlear, and abducens nerves. Remember, these are cranial nerves that are going to control those extrinsic eye muscles. And then we have vestibular neurons that are going to send axons to another specific nucleus in the thalamus. This one is called our posterior ventral nucleus of the thalamus. And from there, we have our last synapse, and the axon will send over that information to the vestibular area in the cortex. So that is the end of our Chapter 16 lecture. If you have any questions, please feel free to contact me.